Oh, really?
Yeah, that would be good.
I think we have these referred to each other and they're Latins, they're both Irish. Which is kind of unfortunate. There was an opportunity.
Oh, yeah. And
Yeah, I could say that one room.
Yeah.
All right. So thank you everyone for being here today. Before I forget, I want to mention that if anybody wants to come and chat more after the seminar, we're going to go to Bombs Away after this. So feel free to join us. It'll just be and so I would like to introduce Dr. Lena Fish. She studied biology and art history at Swarthmore College and earned her PhD in ecology and evolution at Princeton University. She did postdoctoral work at the University of Oregon, our neighbors, and then also at Duke University. She's currently a professor at the University of Montana, where she studies evolutionary genetics in plants, specifically in bee colonies. And her interests include selfish adaptation, speciation, and evolution. So thank you so much, Lena, for joining us. Thank you. Turn it over to you.
Okay, well, thanks, Katie, for the kind introduction and for inviting me, and for her and the grad students for hosting. It's been a lot of fun. It's really gorgeous here. Happy to be back in Oregon, even it's not the exact same location where I've spent some of my formative years. Feel weird with the door behind me, but hopefully no one will speak up. So I'm going to talk today about one of our projects. I work on lots of different things, but I'm talking about our work that we're doing with the yellow monkey flower Mimulus guttatus in Yellowstone National Park. And yes, and before I get into the talk, which I will hopefully move through not too quickly, I want to make sure I acknowledge these folks who've been working on that. So 2 postdocs, Finley Finseth and Tom Nelson, have been very involved in this work. And then Corey Colas was a master's student who a lot of the work I'll talk about is from his thesis. And then Colette Berg is a current PhD student and sort of tangentially involved, but is deeply involved in our ongoing work on the system. And then Peter Bryggens are here. Read is visual on the ceiling, but not on.
yeah.
It went up there.
What?
You were pointing to up.
But it doesn't work on the screen.
He's an undergrad, did some of the QTO mapping. I'll talk about this for an honors project. So I won't point that. So I'm an evolutionary geneticist. I guess now an evolutionary genomicist. I'm old enough that I go back to the era when genomics was a lot of AMLPs, but we are dealing with actual genomes now. And for us, the big questions that we want to answer, I think in general, if there are other questions, but we're pretty much all-encompassing questions, is what maintains variation within populations, especially variation in traits related to fitness. How do you maintain fitness-related variation in populations? And I should say, just because we're nearby, we do a lot of that work up at a place called Iron Mountain. Work I started when I was a postdoc with John Willis at the University of Oregon. Lots of us, John was a sort of founder of the Mimulus community, and lots of us still work up at Iron Mountain. It's just go until you meet the Mackenzie River Highway, and it's at that intersection. And so we work up there on all kinds of questions about how you maintain variation within a population, including things like timing of flowering and fluctuating selection on that, variation in things like herbivory and growth defense trade-offs, and how you might maintain variation in defense traits, as well as a whole set of projects involving selfish evolution, particularly selfish centromeres, which is a whole other thing I'm happy to talk to anybody about, but we've managed to discover in the system, more or less by accident, what looks like an example of the selfish centromere hypothesis for eukaryotic centromere evolution. And it segregates in this nice, beautiful wild monkey flower population. So if we have a lot of variation within populations, we can also then have population divergence, and so this tends to be things about adaptation. And currently we're working on lots of kinds of adaptation questions again, sometimes involving traits like trichomes, but also we're currently doing a lot of work with this really cool trait of stigma closure. So the monkey flowers have a bilobe stigma, and if you touch it, like a bee, if you're a bee and you touched it, it will close like a little tiny inside the flower Venus flytrap. So very rapidly, less than a couple seconds. And then if you didn't successfully pollinate it, it will reopen. But if you successfully pollinate it, it will stay closed. Super cool. We're trying to figure out how and why and they do that, and whether it's a co-option of the general sort of pollen style signaling system. And then I'll tell you today about work we're doing in Yellowstone, looking at a variety of traits. And then of course, if you have population divergence, you might also evolve reproductive isolation. And so a lot of the work that we do slides towards thinking about speciation or looking at the evolution of important traits for reproductive isolation, like pollinator differences or post-mating barriers in pollen tube growth, or things like cytoplasmic bell sterility and other kinds of hybrid compatibility. So this is obviously a little bit of a generalist, or ADHD, I don't know. But I'm really focused today on this, going back fast, on this middle one. Okay, on this middle one, I'm thinking about adaptation. This where the work we're doing in Yellowstone is very focused around adaptation, but I think you'll see that it actually ends up going in both directions, that it ends up being about standing variation in a complex landscape and about the evolution of reproductive isolation as well. Okay. So why do we study the genetics of adaptation? I'm trying to be educational here. I don't know what everybody's background is, but there's really two reasons. The first is to try and understand the functional and molecular mechanisms underlying natural variation. We have 4+ billion years of life on Earth, and there's a lot of solutions to a lot of interesting, difficult environmental problems. Showing you here this mushroom spring in Yellowstone near one of our sites. This is where the Thermus aquaticus organism that produces Taq polymerase, which revolutionized all this stuff, comes from. You never know what out there might be a really interesting solution to the problem of high temperature DNA replication. So there's lots of things we can learn from natural variation that are useful. And then as evolutionary genetics, we obviously also just want to understand the evolutionary processes and understand the forces that generate and maintain variation, and in many cases apply those in like a conservation context. And I'm just giving you an example. This is from my close colleague Jeff Good, who's a similar interest in similar diversity of things, but in mammals. And among other things, he works on snowshoe hares. So out here, you guys would have the brown hares. They are an offshoot of snowshoe hares, which normally turn white in the winter, that stay brown all winter because you don't get a lot of snow out here. And so on the west side of the mountains, they stay brown. There's a certain admixture zone. And so the brown ones are found where there's not winter snow, and the snowshoe hares are turning white in winter elsewhere. They're crypsis against predators. So that's a lynx. And at first they did this work, and I was like, oh, well, you're gonna find it's a goody. They did, GWAS to figure out what the gene was, and it was a goody, which is a classic, well-known gene that affects co-color variation in many mammalian organisms. But what's really cool is that they found that the agouti gene that confers the not changing the brown winter coat actually integrates in from jackrabbits, and is like don't worry about the details too much, but basically this jackrabbit variation is interested in to confer this adaptive variation. So that not only have they adapted, but they've done it through this complex integration, and it turns out it goes even deeper in the sort of balanced polymorphism at these loci across all hares. And so you can really start by getting at the genes, you can start getting insights into really deep evolutionary histories of natural variation. So that's what we want to do. We're not still going to achieve every part of that today, but we're going to try to do that for the Yellowstone monkey flowers. Some more questions, and again, I don't know if you have a core first, these are probably questions you've heard. So when we're studying adaptation, there's lots of kinds of questions we might have that are some of which require getting down to the genes and some of which don't require we get all the way. But first, we're interested in basically the nature of the adaptive landscape. Do organisms have to leap between widely separated adaptive peaks with big mutational steps, or can they sort of walk continuously towards novel phenotypic spaces without passing through valleys of low fitness? So that's the sort of rugged versus smooth question. And then related to that sort of size question, there's also the question of how do you put together complex traits like the selfing syndrome, which we've studied in Mimulus, or floral syndromes associated with different pollination systems like hummingbird versus bee. How do you put together complex multi-trait syndromes and keep them together often in the face of gene flow? You'll probably guess that the answer often involves things like supergenes and other factors that suppress recombination. But that's a fairly new thing that we know that about many systems. We might want to know about parallel evolution. So when you run the tape of evolution multiple times, and you have similar phenotypic outcomes in similar similar ecological circumstances, so parallelism or convergence at the phenotypic level, does that also share a molecular basis? And so are there constraints on the kinds of genes that are involved in a given adaptive shift? And when you're looking at relatively short-term evolution, is the variation come from new, is sort of the selection there and you're waiting on mutations, or is it that there's a lot of variation and then the environment shifts and that evolution happens immediately? So de novo mutants or recruiting from existing variation. These are not new questions. There's lots of answers to these questions in lots of systems, including mice and sticklebacks, and now in Mimulus for the hummingbird pollination. And sometimes does this even go as far as basically taking adaptive variation from other species? So where does the variation come from? Very classic question. This is going back for you plant folks to like Clausen, Keck, and Heise's classic circle transplants of plants. We can measure local adaptation by moving organisms between habitats. And as you can see here, the dark ones do best in their native habitat. That's the classic signature of local adaptation at the organismal or population or genotype level, genotype in the sort of ecotype sense. And when you see a pattern like that, so that's what we call local adaptation in the sort of plant ecological genetics tradition. Is that really happening at the gene level? So is it that there's antagonistic pleiotropy, that a mutation that makes you good here necessarily makes you bad there, meaning that you cannot move those alleles around in the landscape? Or is it that organisms build up those kinds of trade-offs at the whole organism level through mutations that don't have those properties and are permissively spread? So again, these are kinds of questions that we need to get down to the level of the gene in order to answer. And last but not least, we're interested in understanding whether adaptation can cause reproductive isolation. This is a bit of an outdated example now, but there's lots of instances in which people think that so-called magic traits or traits that affect reproduction can influence mating patterns to the extent that you can actually evolve reproductive isolation in the absence of extrinsic geographical barriers. And so when we think about adaptation, we can ask, well, is adaptation enough to create speciation in the absence of some kind of allopatry? Okay, so those are a lot of questions. Again, I'm not necessarily going to answer every single one of them, but I'm going to try to hit on most of them in this particular system, which is monkey flowers in Yellowstone National Park. Probably many of you have seen monkey flowers. Hopefully you've seen monkey flowers. They have their center of diversity out here in Western North America. For somewhat historical reasons having to do with the amazing personality of John Willis, who was my postgraduate advisor. They've become a model system for evolutionary genomics. But the other reason is that they're tremendously diverse florally and also ecologically and in terms of their adaptive capacity. In particular, and I should say, we work not quite across all of this, but through a lot of it, but mostly on the yellow ones that I'll talk about today. So the yellow ones, as you've all again seen, hopefully growing along roadsides around here, out on the coast, up in the mountains, are the Mimulus guttatus species complex. Somebody sent me a question about whether we still call them Mimulus. Monkey flower researchers who are evolutionary geneticists tend to still call them Mimulus. I will call them Mimulus. We can discuss taxonomy later. But it's a species complex, a lot of diversity, exchanging alleles from basically the Aleutians down into Mexico and Baja. And out to over here, which is where they sort of the far eastern edge of the ranges in Yellowstone. These guys are amazingly diverse. They show a lot of adaptive adaptation for the non-planty folks. That means they can adapt to weird soils, copper mine tailings, serpentine soils, just strange, nasty conditions that other plants can't live in. They show a lot of mating system evolution, repeated evolutions of self-pollination, which is common in flowering plants. but occurs over and over and over again, so that's broken off there. And then they also show dramatic life history variation, and I'll talk specifically about this life history variation in a moment. But basically what I mean by life history variation is that there are plants that are annuals because they don't make these stolons or sometimes rhizomes. Stolons are above ground, rhizomes below ground. These vegetative structures that allow them to overwinter. Effectively, they basically go to flower and die, and try to die well, having refused. So this system's been a model for a long time to understand all of these kinds of things. And I've been studying it since I was a postdoc at the University of Oregon many years ago, starting out. And so I was really, when I moved to Montana, I should say I was super excited to see there were yellow monkey flowers all over Yellowstone, which is nearby, and then it's about a 4-hour drive away. Montana's a big state. But so it's on our side of Montana, but still pretty far away. And so there are monkey flowers all over Yellowstone. And most of you, hopefully, anyone never been to Yellowstone? Oh, wow, you should go. So come visit, we'll point you there. So thousands, tens of thousands of thermal features, springs, pools, geysers, all kinds of things. It's a super volcano, so there's a lot of heat underneath the ground that produces 3/4 of the world's geysers. And also bison and grizzly bears and other cool things like that. But from my perspective, and also really cool bacteria that live in these hot springs. I have to mention this because my husband studies the orange stuff. It's the thermal limit for photosynthesis. Very cool. So a lot of really cool biology. These are extreme conditions. Obviously that Taq polymerase came from there as well. So most people think about these things. This is what the tour buses come from, but I come for the yellow. Can't see, it's not as bright here, but Mimulus cutatus is beautiful yellow. And Mimulus cutatus is everywhere in Yellowstone, and mostly in like Montana and Idaho, you'll see ones that look like this, which are these perennials. They live in dense year-round wet thermal bogs. They're under snow in the wintertime. But in Yellowstone, we have a variety of other habitats that are basically some form of groundwater, often heated, that is ephemeral and produces more of an annual habitat. So ranging from all the little yellow bits in the crust to treacherous bogs to these sort of flats that are fed by groundwater. So a lot of very different kinds of Mimulus habitat in this mosaic within the park. And this is really when I first started going there with Scott, my husband. He was a grad student at Oregon, so I started going out to Yellowstone before I even moved to Montana. I was like, oh, Mimulus, it's so amazing. It's so plastic. It can live anywhere. And I didn't really think about it from a genetic perspective, naively, because it is, Mimulus cutatus is very plastic. It can live everywhere. But then once we got out there, I was able to get some seeds from someone with a permit, and we grew them in a greenhouse. And it turns out they're not just super plastic, they're also super genetically differentiated. So there's an adaptation to this complex mosaic of habitats. So just to tell you a little bit about that habitats, and I'm cartooning a bit here. We've worked in very extreme habitats, but it is a more complex mosaic. But I would say at the one end is this perennial bogs. So there's a lot of snow in the continental part of the country. So this is in May. It's high elevation up there. It's under snow until May, June. Some years I've been up there in like a massive snowstorm in mid-June. But then eventually it stops snowing. Plants come out from the stolons and the bogs, they flower, they're pollinated by mostly bees, bumblebees, and then they're done again by September when it gets cold again. So this is the one extreme. The other extreme is, and this is about, I would say 200 meters away from that other picture, is a variety of thermal habitats, but this one is a thermal habitat. The most extreme ones are completely snowmelt fed. So this is a place we call Agrosta's headquarters. This is in the canyon, and you can see in March, there's tons of snow. In April, they'll melt it out again. It could snow again. The snow melts when it hits the hot soil. So it doesn't look like anything. There's no pools there, but that soil, if you stick your finger in it or thermistor, it's hot. I was like skeptical at first. I was like, this doesn't look like Mimulus habitat. There's no water. Yellow monkey flowers require water, but they're living off the melting snow. And as long as they stay super close to the soil surface in January and March and April, you can see here, still tons of snow in the background. This is all thermal area. As long as they stay closely pressed to the ground, they can live in this tiny boundary layer of like pleasant soil, pleasant air, high humidity, good moisture. But then, by June, if it stops snowing, it's hellish. the soil is dead. And you can really, the moss here is your clue as to what's going on. So the moss is green when there's water, the moss is not green when there's not. And so everything is crispy and dead. So this is an obligately annual population. Nothing lives for the summer there. The moss regreens. So you guys have to be annual, set seed before the summer comes, the soil dries out and they die. And so they do this by not by tolerating the summer. So I suspect that there is some thermal tolerance going on. We're working on that as well. But what they're doing is and this is the classic Mimulus cutaneous trick. I think it's a general plant trick, is they're avoiding the most extreme. They're very, very evolvable in their life history. So what they're doing is they've flipped their entire life cycle to basically germinate in the fall. Snow is falling, melting on the soil. They are out all winter long, exposed, snow-free. They'll freeze if they get tall, but they stay close to the ground. And so like here in January, they're out photosynthesizing. Can't see it, but soil temperatures, perfectly reasonable. Air temperatures above the plants get, into the minuses, -20, so cold. But as long as they stay close to the ground, they're fine. And then it gets, warmer, and starts peaking out here this year. It's over 45 in May. You can see it's over 60 in the summer, and they can eat then. So what they've done is they basically have switched their entire life cycle from being summer to being winter, but they're taking advantage of this beautiful, low competition, lush habitat that's only available to plants that can live and flower and reproduce in that window of time. And there's 2 species of grass that live in this habitat and no other vascular plants that do well, except for some weeds that are coming in now that are pretty adapted to this. Okay, so it's not really a story of thermal tolerance, but of evolving your life history to fit this window that's out there. Okay, so this is data we actually got much later than a lot of the work here, but just to say that when we bring plants in from these habitats, this is from all across the park. So this is both extreme annual and perennial, but also some intermediate sort of wet all summer, but probably dying by August kind of habitats. We define plants as sites as annual if they're toast in August, and as perennial if they're still wet in August. That's the simple. That's all we're dividing plants by here. It's much more complicated than that. But you can see that they don't fall out perfectly, but there's a strong axis of allocation to flowering branches versus stolons. Those are the overwintering structures, and a strong axis of mating system here, which are the two primary things. So in order to be a good annual, you need to basically allocate to flowers rather than stolons, and you need to be able to reproduce in the absence of pollinators, because when there's a lot of snow around in April, like that's not a good time for bumblebees and meals. Okay, so they're not out there. So they become very good at selfing. And there's also a number of other traits. We've done a whole bunch of work looking at the genetic basis of early trichomes on extreme annuals. There's one of the grasses there you can see as well. They become super furry in the most extreme annual sites just while they're young leaves. We do snow shedding. This is work that Margaret Hendrick did for her master's. And then my current student Colette is doing a massive GWAS to look at, she's already completed the flowering phenology, super high levels of variation in critical photoperiod for flowering, which is basically the day length cue you use to decide to initiate buds, with some flowering under 12-hour days and others in the non-thermal only flowering for 15-hour days. So that's like the difference between March and summer, June, July. And she's doing a massive GWAS on floral mating system traits. So I'm not going to talk too much more about those here, but focus on those life history traits that are really the key thing that distinguishes the way you can live in these two habitats. Okay, so basically, as I said, we're going to focus on the life history. We have sort of this perennial world, and it's not just that it's consistent. This is a high competition habitat. So these plants are living in a sort of typical bog. Sometimes there's actually some thermal influence, but it's a lush summer typical plant Mimulus habitat versus this annual world where they've evolved to live in the wintertime and then know that death is coming. And so everything's about getting your fruits made with seeds before the inevitable summer. And so, and we're interested in this not just because it's a cool thing of adaptation to the site, but because annuality, perenniality is a kind of important trait in general. Life history and traits in general are very important. But it's a common pattern in plants that sort of weedy or ruderal things evolve annuality. If you are knowing that you're going to die from extrinsic forces, then you want to be good and die, reproduce and die having already done all your business, versus going for multiple years. So basically, bother with this. So these life history traits, they control the timing and packaging of reproduction, and in plants that allocation to basically vegetative reproduction or growth, so making these stolons or rhizomes, little side shoots, versus basically only making flowers. And so in our populations, the annuals have lost the ability to make these, and the perennials continue to make them. And this trait, these kinds of traits are actually a really important component of the domestication syndrome. So if you know about this, is a whole suite of traits that basically move a lot of crop plants from being things that, put resources, store for the future, versus things that basically put everything into some kind of fruit or seed, grains, things like that. So many of the things this is just a review of domestication syndrome. Many of the traits here are about shifting allocation from basically asexual reproduction or growth into yield or seed production. So it's really interesting, but despite this being a big component of domestication in many crops, it's really, for some reason, it's not been well understood genetically. And I think it's mostly because of Arabidopsis, the model plant for a lot of molecular biology, is a strict annual. All its relatives are perennials, and they are not cross-compatible. So no one's been able to do the big QTL map. So there's a lot of interesting questions about this that just haven't been resolved in any system at all. And in particular, we want to think about how do you put together strategies, like is that axis of reproduction, flowering versus stolons, is that a necessary axis, or is that loss of one and gain of the other separately? So is that a continuous trade-off? So is there some kind of intrinsic trade-off where you're using your meristems, you're using your resources, and one switch gets you both things simultaneously? Or could you have it both ways? Ideally you could. So, and actually, some of you may have heard a lot of what is known about the genetics of life history variation, annuals versus perennials, known from Mimulus guttatus. In particular, this is work by first Megan Hall and then Dave Lowry in John Willis's lab, working with plants from Iron Mountain, just east of here, south, and from the coast in Oregon. Let's start looking at. So IM is up there, Dune on the coast, SWC is in the coast range. So Megan started working with this, and she did QTL mapping of annual versus perennial. They're totally crossable, not different species, despite the taxonomists might say. And got this locus div 1 on chromosome 8, where basically each of those colors represents different traits, all the traits associated with these differences, stem thickness, flowering time, leaf size, making of stolons, it all maps to the same place. So all of these traits go to this locus called div 1. And then Dave demonstrated that there's a big inversion on chromosome 8 that controls this, that basically thousands of genes in this region. And then there's another inversion on chromosome 5 that together basically are a set of variation that condition you to be annual versus perennial. But each of them is, again, hundreds, I think thousands in the case of genes. So there are large numbers of genes. The inversion prevents recombination in that region in heterozygotes. So pretty typical super gene story here, but one of the first ones. And if you go and look at the things across the whole range, the different colors are different clades. In each of those clades of Mimulus guttatus, these two loci define annual and perennial, but otherwise the genetic structure is primarily geographic. Okay. So, and another cool thing about this is that and which provides a nice sort of frame for what we want to ask. These are very typical annuals and perennials, basically live fast, die young, make stolons, grow slow, get big later on if you have a long growing season. And we see that, just walk over here, we see that both in wild accessions, the slower you are to flower, the more stolons you make. So that's on the landscape, but in a common garden. So that could be the same genes or not. These are F2 hybrids, so recombination breaking up. But still a strong genetic association between number of stolons and days of flower. So that basically you can be stolon-y and slow or not stolon-y and fast. And that's the classic sort of life history trade-off we'd expect if these were under joint control. So what about Yellowstone? And so one of the first things we wanted to ask for the system is, we want to know how they do this. We're pretty sure it's directional and it's derived, but are they doing it the same way? So the first, and I should say, and we're going to answer many, many questions about that. I'm not gonna read them again, we'll answer them as we go. So first question is, the first obvious thing is like, do they reuse the same widespread things? We're way over there on the edge of the range, it's kind of a special set of circumstances. It may not be typical annuals. There are annuals pretty close, but maybe not right up next to Yellowstone. We tend to have mostly perennials in the surrounding regions that are not in the park. And I should say, 6 months ago, I would have shown you like a whole ton of linkage mapping and a lot of indirect information. I don't have to do that anymore. We have the genomes. So we can just tell you this question. So as part of this really large JDI project, we basically sequenced a thermal line from Yellowstone. We're in the process of sequencing a non-thermal line, but we've also sequenced lots of our other reference lines, like the one from Iron Mountain that we've been working with for years. So now we have beautiful PacBio and Hi-C, basically telomere to telomere chromosomes. And what we can do with that is just line them up and be like, which order do they have, annual or perennial? And the way you read this is just a dot plot. So these are annual. It's the annual order here. That's our original reference. And then plotting the HQT, thermal annual, against it. And you can see, hopefully, this is an inversion on the end of 5. This is an inversion that corresponds to div 1. So both of these have the perennial non-annual order. So, the perennials in there do as well, but the HQT or thermal annuals, so our flagship population, are basically perennials chromosomally. Okay, so they could be doing with the same genes, but they're not doing with the same chromosomal cassettes. Okay, so novel genetic basis. So then we wanted to say, well, what is it? Okay, it's not the same thing. Is it another big chromosomal inversion somewhere? Is it one gene? What's going on? And again, this is work that sort of accumulated over the years. So we started with a pool-seq approach in 3 focal populations. One of these is at this place we call Agrostis headquarters. It's where the pictures I showed you earlier from. This is our most extreme pair, separated by a couple hundred meters max. The canyon in here is mostly thermal. The extremes have FST about 0.2, which is pretty high, very high, be a species in a bird at least. And then we have this other site where there's this hot creek that is home to mimulus that are in a thermal area and die, but flower all summer. And they have extremes up the top, but it's a much more connected landscape. And here, FST between the thermal and non-thermal extremes is relatively low. So we did pool-seq of all 3 of these pairs. And we're basically just asking the question, because we want to find the thing that causes annuals to be annual, perennials to be perennial, what is shared between these 3 sets of pools? And the reason we have to do all 3 is that, everything over in HQT is highly differentiated. So we do all 3. Hopefully, this is just a classic pool-seq. Lots of seedlings collected from the field, independent moms go into pools, sequenced into high coverage. And we're just asking the question, where are T and N always different? And again, T and N here it's really annual and perennial, but it's thermal annual, non-thermal perennial. Okay, we do that, and we actually there are things that are differentiated in each pair of pools, and there are things that are genome-wide differentiated, but there's basically one region of the genome that includes about half of the highly differentiated sites. And this, whatever metric we want to use, whatever analysis we want to use, but there is a 20 gene region on the end of chromosome 6 that we call OUT6 for outlier 6 that basically is, holds half of the outliers for the whole gene. There are a few other really interesting genes that are single gene outliers. These are easier to get at functionally because they're not 20 genes. I'm not going to talk about those today, but we're going to focus on this OUT6 region, which again has all the properties of those 2 big chromosomal arrangements that distinguish the types that are found all across the range elsewhere. Okay, so what is OUT6 doing? And I should say that we found this through pool-seq, which is just wild plants collected into tubes. For all we know, it's involved in root heat shock. We don't know. So we have to connect it to a phenotype. These are pools by location, not by phenotype. So to connect it to phenotype, Peter, really a great undergrad, did a QTL mapping. We were F2s between our HQT and HQN extremes. Remember, they have high FST, so we can do this. We have markers genome-wide. And if we do that, we find that this OUT6 region basically is the big locus for stolon count, which is, again, stolons are the perennial trait, and also for internode length. And so annuals our thermal annuals are quite short. So, and so there's multiple traits that are wrapped up in this region. We can also, in this situation where we're sort of breaking correlations at least down to the region, we can look at that same, this is the same exact graph I showed you for the widespread ones, and here we see the exact opposite pattern. So the more stolons you have, the faster you are to flower. Okay, so this is annuals are living slow and definitely dying. I don't know if they're dying young, but basically what we have in Yellowstone is a very different kind of annual strategy. You can see these are annual genotypes, height and flowering and other things. So these are actually, for this particular cross, basically, annuals are hunkering down because remember they have from like snowfall until summer dry down, whereas perennials have from when the snow melts out in May or June or July till the end of the summer. And so again, this is a situation where there's, there could be, but there's not an intrinsic trade-off. You can be basically a fast perennial or a slow annual because that's what natural selection is favoring. So I should say that there's a lot of flowering time variation, and this may be particular to this extreme site and not every single region park, but we probably have some fast annuals as well. Okay, so far I've told you that we have this really extreme geothermal habitat. This is a picture in January. You can just snowshoe in, snowmobile, and then snowshoe from Old Faithful from the lodge. These plants are out. I think the moss is probably really important in their ecology. We haven't quite gotten into that yet, but all the little rosettes here are totally out, photosynthesizing. And again, air temperatures, my height are, frostbite, but they're in this little sauna right next to on the ground level, and are, as long as they stay in there, are great. Sometimes we see actually like cold burn on the leaves, so I think they're actually exposed to more cold stress than heat stress. So, given this really, really unusual extreme habitat, they've adapted to it using a novel genetic basis that from this sort of perennial chromosomal chassis. So it's evolved independently and locally. We have a new locus, out6, that controls this and lots of different aspects of plant architecture. It's only about 20 genes long, and it does the things it ought to do by natural selection rather than be constrained in any way. And again, that just really emphasized that, a lot of these things that we see over and over again as replicated patterns across populations or across species, maybe because the world is made up of very distinct kinds of places. And generally speaking, when you are going to die for sure in a Mediterranean habitat, you have a short growing season. And so that sort of live fast, die young goes together, but it doesn't necessarily have to genetically, but it does ecologically in many cases. But this is sort of an exception that shows that it's actually built by natural selection. So that's so far. What I want to do now is dig a little bit more. So we have this region that does all these things that the big chromosomal rearrangements did, but it's a relatively small region. And we've sort of found it in these few focal populations, but now we want to sort of ask, well, what is it? Where does it come from? What is it doing? All these kinds of things. This is work that Tom Nelson did, population genomics, and I can't really point. So we're looking at the end of this out6 right here is a little red area. And these different lines are basically various measures of diversity on, this is only about 8 to 10 lines. We're working at about 90 now. It will hold. But sort of example lines of inbred lines we made early on. And basically we can see here is, this is chromosome 6, this holds for the rest of the genome, is that the T and the N, sorry about the colors, they change sometimes, but blue always means N or non-thermal. And the hot colors are the thermal, so red, yellow, orange. You can see that there is low divergence. So D sub A and I between is low everywhere. Again, these are populations right next to each other in a complex habitat matrix. And diversity is variable, as you'd expect, but similar, because these are basically the same plants everywhere else. But then when you come into this out6 region, divergence becomes very high and diversity goes down. Each of them is basically a single haplotype. Okay, so we have low diversity and high divergence in this region. Like with the hares, we can build trees with this region. And basically, these are lots of other sequence stimulus from across the range. Annuals are in yellow, perennials in green. You can see those, but you can see they're intermixed, as you would expect. You can see that these are very, very divergent haplotypes. Okay, so a lot of sequence differences. Michael Nachman once was like, are there any non-synonymous differences? I'm like, yes, every gene has dozens of non-synonymous. So there are a lot of differences. But then as soon as you go outside, the thermal, these are the Yellowstone ones, and, geography is what matters. Okay, so this is something that's happened locally, but this is a lot of, accumulated variation, but not a lot of accumulated variation within each, okay? And so we can use those patterns to then sort of answer these questions about where the variation comes from. And so first thing is that it's possible that it's a very old de novo mutation that has some history elsewhere, but given how it's not like a single SNP or a couple of coding changes, there's a lot of differences. And so we think this is probably old variation that has been recruited for this. We don't know exactly where it came from. And in this case, we don't have a telltale signature integration where the haplotype at the annuals matches some other plant. There's an addendum caveat to that in a moment. So it looks like, so whatever variation is segregating here has a much deeper history than the current populations, which again, the haplotypes are very similar to each other. So there's not a lot of variation within, suggesting relative recency. Okay, so the other thing we can ask is about antagonistic pleiotropy versus conditional neutrality. And again, this is the question of whether we see signatures consistent with strong ongoing selection for one type here and the other type there. Again, it's possible that we could have found that, say, non-thermal perennials, which we think are the ancestral type, are very diverse, and that the annuals have went through a bottleneck of adaptation onto those thermal sites. That's not what we see. We see both of them showing really strong signatures of recent selection in terms of their low diversity. I'll show you some more data that's relevant to that far slide as well. So this pattern of reciprocal sweeps then also suggests there's sort of strong ongoing current selection in the face of ongoing gene flow between the habitat types. Okay, so that's just sort of constantly every generation we pick annual here and perennial here. And these particular haplotypes. And that suggests that each is bad in the other. And I suspect it's a little, well, we can go to this more if you want to check, but I think it's a little bit asymmetric, where it's really bad to be one on one habitat type and a little less bad to be the other. Okay, but we want to sort of, again, it's all been done in a relatively small number of lines or the pool seeks, which are these focal populations. And so my student, Corey Colas, did an amazing amount of backpacking and fieldwork. It's with these sites in Yellowstone, we can't just go to any site anytime. So the thermal areas especially can't be there with tourists are there. So we spent a lot of time going to the park, like when plants are not flowering or just died, because we can't be visible near boardwalks, like off boardwalks, except like if a ranger is standing there being like, it's okay, they're doing research. So it's very constraining. So we basically had to get into sites whenever we could. And so he backpacked all over the park, as well as did a lot of collecting in the boardwalk areas, about 300 different samples across, again, a much more complex mosaic than the sort of extremes I've been talking about. And again, defining sites as annual perennial, that's the color. You can see all the geography includes both annual and perennials in each site. And then he grew up, because we had to rescue plants from the field and grow them in the greenhouse, we took self-progeny to do common gardens. And that's the data I showed you earlier. But again, just to remind you, now we know about OUT6. And so we can ask, okay, we know that there's phenotypic differentiation. We know that we find this locus that seems to condition it. It acts as a QTL. What does it do on the landscape? And you can see here that it is strongly associated with habitat, not surprisingly. So again, hot colors being thermal. We find lots of those alleles at annual sites, fewer perennial sites, not very many heterozygotes, given, which is a little surprising actually. And then if we look at the phenotypes, again, in a common garden of plants from all these places, there's lots of different phenotypes, but if you look at stolon count and your genotype at this locus, we have markers in it. Again, super explanatory. And I should say this is a little bit unfair, and why we did the QTL and the pool seek we did is if I were to put stigma separation, which is a mating system trait on here, which we know is not associated with this region in a QTL map, when we break the linkage equilibrium that exists in the linkage, we would see a similar pattern. So OUT6 genotype is highly correlated with habitat, and habitat is associated with lots of phenotypes, but only some phenotypes are directly causally associated with this locus. So, but in this case, we know it is QTL. Okay, so this again is consistent with what I said earlier from the sweeps, which is that there's really strong selection, sorting variation at this locus on the landscape all the time. And that there are other phenotypes like mating system and height and things like that that are associated, but they're sort of ecologically associated. And sometimes you could be a tall annual and sometimes you could be a short annual. and it depends a little bit more on habitat, so they're not totally, it's not conditioned by the same locus, and they're not totally co-linear in this phenotypic space. Okay. The other thing we can ask is, so we have this 20 gene region. I said that their haplotypes are basically identical across the whole thing. This is the bane of my existence. I always want to break things up. Like cystic liver is our friend, but it's also our enemy. So we want to know, like, is it all 20 genes? Is it one of the genes? Like what's going on here? Is it selection on both ends of that interval? Why do we have this whole chunk that's moving around? And so, again, being a person who processed things, I went ahead and made over 1,000 F3s to try and get some recombination. I didn't get any, meaning I couldn't get any individuals that broke L6, where it was half and half in any of the alleles. And we also didn't see any recombinants in about 300 wild plants. So that is suggestive of something like an inversion. I mean, there's a lot of interest in these sort of supergenes and how you get them. But I should say we know that there's no, if there is an inversion, it would be distinguishing the perennials from everybody else. And they're the ones that we're still sequencing. I started with the annuals. But there's no evidence so far, and we've looked a lot at all kinds of sequencing data short of long reads that actually span the interval. So I don't think there's an actual inversion, but I think it is a general recombination cold spot, because this is actually, it's right at the very end of the chromosome. There's like telomere sequence within like a couple kb of the last gene. And so I think it's an area where there may be, combined with selection, there may be a low enough rate of recombination this is holding together, despite a fair amount of gene flow. But it could be an inversion next year, we'll see. So what is actually in there? I don't know if there's any molecular biologists here that want to know what's actually in there. It's not a goodie. So this is the region, and the numbers, those of you who are very sharp eyed may notice that our numbers changed. This is our new assembly that is from our thermal line. And so now we're aligning to it rather than to the annual organ reference. And I should say the most important gene probably is this one here. The very last gene on this chromosome is gene CLAVATA2, which is a key regulator of meristem proliferation. It's single copy essential gene in all flowering plants, pretty much, which is why we're not trying to break it, because we will have phenotypes, but they will not tell us anything. If we break it, the plant will not make meristems. But then the next two genes over are tanoduplicates of PIN5, which are regulators of auxin, auxin being the major plant hormone associated with branching patterns. And then the gene over on the other end of the interval is in this PP2C family, which is, again, loosely related to things involved in meristem regulation. And again, in Mimulus, we don't have like the knockouts you have in Arabidopsis. So we have to use inference to be like, okay, this gene does X in Arabidopsis. And so maybe it does something like X in Mimulus. But any of these are good candidates, and we don't have any recombination between them yet. Just to show you how frustrating that is, this is sort of plant gene breaking studies, wild type sorts of studies where people have broken PIN5s in rice here, and you can see that the wild type makes side shoots, something like stolons, and the mutants don't. So you can do it. And then this is, again, a lot of different things here. Clavata is involved in a lot of things. It's not a gene you generally would want to mess with. And again, it's involved in things like tillering patterns and growth form in Arabidopsis. So we still have multiple, even them down to like not that many genes, we still have candidates. So we're still trying to narrow down that because we always want to try to figure out, I mean, is it that you need to have both ends of it, or is it just, carry along? Why are we seeing the same region, the whole region? I should say we've done RNA-seq, annuals, perennials, nothing tail. We haven't gotten down to the finest meristem scale. We're doing a huge GWAS that will involve all the floral traits, but also more variants, new genetic variants that we can ask about all the traits on. That should be great, but it might tell us something more about ALK6. And then fairly recently, we discovered this other thing, which hopefully will be super useful, but is also frustrating in the nature that biology can be, which is when we did that screen that I showed you that Corey did of ALK6 haplotypes all across the park, we found a rare third haplotype that seems to confer the exact same phenotype as the annual haplotype. It's like less than 5%. So this is not what I expected. Again, I expected the perennials to be diverse and the annuals to be like the one good way to do it. There seem to be two totally distinct haplotypes one of them quite rare. And that one is clearly integressed from this other species, Mimulus micranthus, that's supposed to be restricted to the Coast Range of California, but through some flicker prospecting, I discovered there were Mimulus on rocks in the north side of the park, out of the thermal areas, and Mimulus micranthus is growing up there. Tiny flowered sulfur, so it looks like in the greenhouse. It's actually a very robust plant in the greenhouse, but it has the exact same, a couple SNPs, but it has basically this third haplotype. So that one clearly came in from through integration, and so there's definitely something more complex and weird and complicated that I have to figure out before we get done on this project. So, but the upside of that is that because these two haplotypes confer pretty much the exact same phenotype, and they're quite divergent, we can now start narrowing in. There's a region actually sort of interior and a couple of chunks right upstream of Flavata 2 that look like they're convergent between those two haplotypes. So it actually is helpful to us to have that, even though it's frustrating from a neat evolutionary story perspective. Okay, so maybe coming down on time. So yeah, so the last question that we want to ask about these variations, we're seeing a lot of evolution in life history traits, in floral traits. I'll show you a bit more of that just in a quick way. And so this is a situation where if there was ever going to be sympatric speciation, like we have incredibly strong selection for plants to basically live in the winter versus live in the summer, right next to each other, no geographical barriers, there's selfing, outcrossing, bee pollination. These are the big differences that are supposed to be the things that could, if anything was going to jumpstart speciation without geographic barriers, do it. So we wanted to basically look at the patterns. I've already hinted that there's a fair amount of gene flow. We wanted to look at those patterns park-wide and see, well, what are the patterns that we see in terms of buildup of reproductive isolation? And this is work that Corey did. And just to show you, this is again from that big throw out. Lots of traits are associated with ALT6 genotype. None of this one, the reproductive branches and first flower node, are associated with the QTL. These two are just correlates of your genotype at this locus. So this one locus predicts your inbreeding coefficient because it predicts how extreme an annual habitat you live in. So if you are a TT genotype, you have more history of inbreeding because you're living in this habitat. You're adapted to this habitat where you have to flower in the winter. And so the alleles are moving out, but they tend to be found in these, you can actually predict those inbreeding coefficients because they tell you about the ecology of the site. So because of that we might think that thermal plants, because they're inbreeding with themselves, might be spiraling off into becoming new species if this could. And don't worry too much about the details. These are a variety of population genetic things. This is your classic structure plot. And as you can see, there's not a lot of structure to it. What structure there is strictly geographic, with one notable exception, which is this HQT population here. And I'll just show you this in the FST world. So that was sort of continuous how we split variation up in the landscape. This is now defining geographic populations, which are clusters. 4 of them are geographic. Moderate gene flow, FST in the 0.06, 0.13 range between them. And then if we look at thermal and non-thermal within each, very low differentiation. So again, it's a big park, there's mountains, there's some geographical structure, but you may notice this little purple dot here. That is the population where we started. It was the first site where we worked. It's this Grostis headquarters thermal site. It's the one where there's basically a hillside, heated, but no water source. And because there's no water source, when the snow stops, the site dries out and becomes 65C. Whereas where there's water, you can start early, but you can also go later and you don't die until much later in the summer. So, this is sort of just one population, but again, this is the scale of it. This canyon is mostly thermal, extreme thermal up the top, and the water doesn't start till about right there. And that's the non-thermal site. So a couple 100 meters. And when we compare basically the top of this canyon to the bottom of the canyon, FST is like, again, 0.19, but FST between HQT and all other populations is always quite high. And so this one population has kind of started spiraling off. And I should say it's among the most extreme ecologically, but it also has this gap. It's here, where again, in the canyon even, they're flowering in April. Again, there's a lot of snow still around. And you can see it's starting to get dry and crispy already. And then down at the bottom of that canyon, these plants are sort of laying down like this because they were just under snow. So they're starting to come out from the snow, and there's probably some overlap in some years, but almost no overlap in flowering at all. And so it's that little bit of geography and that lack of intermediate habitat that creates the barrier on top of the genetic divergence in their and adaptation in their reproductive biology. So, and I should thank Sally Otto talking about the speciation. She was like talking about the perfect storm. You need the perfect storm to get speciation. No one thing is going to be enough. Geography and ecology might get you there. Okay, so to conclude, I'm here. So complex life history traits, again, in Mimulus can have a very simple genetic basis. This one's even simpler than those big chromosomal rearrangements. We have novel solutions to this existing problem. I don't know whether that was adopted because of the unique ecology of the site or because there just weren't that the annual variants of that are widespread weren't available, those chromosomal inversions. But whatever the reason, it recruited some form of old variation, and in a minority case also appears to have had integration from a selfing rock specialist species. This really complex set of traits is maintained on the landscape in the face of ongoing gene flow. So we end up with this really complex mosaic of habitats, but again, consistent sorting every generation, selection, refavoring the match between habitat and genotype. And that hasn't led to speciation across the board, but where we have big enough gap and really strong selection, we're starting to see that happen. And I should say, I also have some very data I don't really choose to believe, where it shows some degree of post-zygotic reproductive isolation involving between HQN and HQT. But so that we may actually be starting to get solidification to the point that they can't mate rather than just they don't mate. But right now I'm of the mind that if a hot spring were to appear at the top of that canyon and flow down, it would all just, would be adaptation between flow again. Okay, so to close, just lots of folks to thank. And I think there's a time for 1 or 2 questions. Go ahead, there.
Okay, so you mentioned the out-6, right? You don't have evidence for introgression.
Well, so we have these really distinct haplotypes,
Right.
And the perennial one, I mean, again, there's a lot of variation in the middle. So any one of them, any one gene in that interval is not necessarily any more different than any other pair of genes between any other pair of populations. We don't find that exact haplotype in any other species where it would be like, yes, it came from you, except in the case of that rare Micranthus one.
And you think that Micranthus is an annual?
Sorry, Micranthus is totally an annual.
Okay.
It's an annual
Does it
cell
have
with
the
tiny
dip-
little flowers that
Does
lives
it
on
have
those rocks.
Does it have the dip1 inversion?
Hmm?
Does it have the dip1 inversion? Micranthus?
That is a good question. I have been suspicious that it's actually a different perennial annual.
Okay.
But that one has been sent to JGI for, so that would, I because it's so robust as well. I mean, in the field, it's tiny, looks like Nasudas, but when you grow in it is a very robust and relatively slow annual. But it's also possible that it has the annual div1 and inversion 5, but that it has additional things that make it especially Micranthus, and those things are useful in Yellowstone. So so it could be either of those, but I am checking on that because I have become suspicious about it because it is a weirdly robust in red chambers. Yeah, good question. Yeah.
I was trying to catch a punchline and might have missed it. So is this an example of sympatric speciation?
So I would say no, but so I would say it depends on what your definition is. So is a 200-meter gap sympatry?
Well, I was just wondering, why does evolutionary biology only look at distance and space but not time?
Well, in time.
That hey, you're always flowering in June, you're always flowering in July.
Yeah, but a little gene flow goes a long way. That is the cardinal rule of evolutionary biology. It's like doesn't take a lot.
Both space and time. So would you consider time to be a strong barrier?
Oh yeah, time is definitely the barrier here. But without the space, I don't know that without that space being empty of mimulus that flower in between them, right? Because if you flower in April and but you stop in April, but somebody in the middle flowers in April, but also flowers in June, your alleles can go down and actually I should say that canyon population has a lot of admixture in it. It looks very thermal. We haven't figured out how it's ecologically different, but like we see the allele, there's definitely, it's not as clean. It's experiencing gene flow because sometimes in a good snow year, when snows till June, it can start overlapping with the bottom. So the top doesn't overlap with the bottom, but depending on the level of intermediacy in the middle, there's a bridge to gene flow. So you don't have to mate with it. You don't have to be mating. So the key is not having mating between the extremes, and that means no bridge. But if there's bridge, which there is everywhere else in the park, all those hot, like swampy things where plants can flower relatively early because it's hot, but they can also flower relatively late because there's water. Those ones will overlap with both the extreme thermal and the extreme non-thermal and act as a conduit for neutral gene flow.
So the paradox is that you have speciation despite all this crazy.
Yeah, so the paradox is that at that one place, I think it's a sharp enough gradient and enough of a gap so that space and time together create, what you need is for speciation is no gene flow. Space is a great way to do that. In this case, it's that space and that space being empty of plants of that species that flower in an intermediate time. That makes sense. So and the time, so space and time, but it doesn't really matter. I mean, obviously that amount of space wouldn't be enough if they all flowered at 2 and the bees were out. But it also wouldn't be enough if there were plants in the middle that flowered and overlap with both. So people often talk about phenology as being a good barrier, but again, a little gene flow, like it acts as a bridge. And so that neutral stuff just go back and forth. Does that make sense?
I think so.
So I personally don't feel that sympatric speciation is possible because it's the existence of gene flow. It's the cessation of gene flow in the face of gene flow, and maybe in some birds, we just stop talking to each other. But in these guys, like it's really hard. It's really hard because it's even flowering basically in March and June, and not overlapping is not enough if there's plants that overlap with both.
That's the punchline.
Yeah, so it's the lack of overlap that is the key. And in this case, it's created by a space that is not hospitable to any plants, but those spaces are all out there in the park elsewhere. And so sorry, do you want me to come down more? I would say there is no such thing as sympatric speciation with breast. But polyploidy is always a thing. But everyone ever likes to talk about those probably like happens all the time. Yeah.
What happens to you if a visitor sees you hiking and looking at monkey flowers?
So it's okay. So when we're in the park, I didn't put it all on it, but we are extensively permitted and we have to wear the special vests, we carry our permit. And so, but we're not allowed off, like that's what we cannot go into like a thermal area next to the kid. Like I have samples from around Old Faithful that I go in April before the park opens. Just super fun. There's grizzlies in the parking lot. Everyone's scary. Like just walking around. So we can go in there like for 1 week before the park opens. The road is open and you can get in, but it's not open to tourists. And then we did some sampling, Corey and I, with the ranger next to us, but like we had to sample like because we're like, what about those nice plants? She was like, no, these ones. And so we can't just walk anywhere. If we're out of the main like tourist areas up in the mountains or whatever, it's kind of a free for all. And we can walk wherever we want. We just have to carry our permit. And if someone like, I was able to say about my husband a couple weeks ago, and I was walking out of a closed area, and someone was like, "You can't be in there." And I'm like, okay. So people do check, like they do a cost. So we try to be very careful and very respectful. We have a lot of trail cameras that we put to monitor phenology, and so we have to hide those. They're not real visible. Is that
I mean, I was mostly wondering because we have people that do work on Mount Pisgah in Eugene,
okay?
and they were the best, and they did the whole thing. But I've seen them get accosted by dog walkers a lot.
Yeah. I mean, this happens up at Iron Mountain because there's a real popular wildflower trail. Like the site is like within sight. Of the trail. Like, what are you doing?
Yeah, actually.
But I mean, yeah. We try to explain we're doing. I mean, I'm working on trying to get some of these weird reasons. I don't know why there's not a lot about monkey flowers in like all the park materials, even though it's amazing. I mean, piece of adaptation is not as amazing as like the thermal photosynthesis. But yeah, so we're trying to get some things out there so people like know about them as a component of the diversity there. I think a lot of it is that the most extreme ones, they're dead by the time the park really opens up to visitors in June. And so people don't see the little brown bits. But I'm like, oh, fabulous. Because they're often dead by the time people are out in the extreme periods. Other question?
Considering that inbreeding coefficient, have you found if you take those, that mimulus that's so adapted to the hot
Mhm.
springs, and bring it to,
And like do autogamy tests on it.
yeah,
Yeah.
and have you found if like it does become more perennial, or if
Oh,
those stolons grow once you take it out of that really specific habitat?
So there's certainly G by E as well, so yeah, so in your genotypes, but yes, like if we grow plants under short days, we can, I mean, I can make pretty much any plant perennial, even like the most extreme, like tiny skinny Iron Mountain annuals, by like giving them 12-hour days, they'll turn into cabbage and like make rosettes in a pot. Like, yeah, so so there is a lot, and I think some of the growth form stuff like the height, the internode length, I think that is quite responsive to air temperature. So we see it even under greenhouse conditions, but it's much more extreme in the field. So they're super short in the field, the thermal annuals, and the other ones are super tall, but that persists in a greenhouse, but it's much less extreme. So I don't think the annuals are very good about not growing into cold air. And but they're probably not very good at responding to competition by elongating either. So there's more, yeah. This is why we've made the inbred lines for doing the GWAS, is then we can do them under different conditions. So far we've just been like one common garden, but if we have permanent things, we cross them by each other, we can do big grow outs under like all kinds of conditions. But yeah, I mean, I didn't really say this because I wasn't talking about the mating system. They are better at selfing in the greenhouse. So they, like the most extreme annual, it's like it sets as many seeds if you leave it alone as if you hand pollinate it, whereas the other ones don't. So, but that's cleansed. That's not quite done like that yet. Other questions? Go ahead.
Yeah, I was kind of wondering about gene flow by pollinators. Obviously, your selfing annuals that don't have pollinators don't have to worry about that too much, but your perennials must have really long distance gene flow going on from long distance pollinators.
Yeah, but again, I mean, generations, yeah, so the stepwise it gets you there. So these FSTs are like integrating across, whatever, hundreds of years potentially. So it's not that they're mating with a plant 20 miles away right this minute, it's that they're mating with the ones down the stream and they mate with the ones further down the stream till you get the Firehole River and then everything spreads, so I don't know that they're super long distance movements, but most of these guys would be pollinated by large bumblebees. And I've tried there's folks who've been doing like, lots of insect capturing in the park, and I've been trying to figure out if we can get really good phenology traces on those. Haven't succeeded yet. I think that project stopped, but yeah, I don't know exactly when the bees like first emerge, but there's a period of time where there's basically nothing, and there's a period of time where it's like you might get visited by a large bumblebee. I should say though, I've worked up at Iron Mountain for years, and I haven't spent that much time in the field, but I spent like whole seasons like sitting next to the plants, and the population is like 95% outbred most years. Like if you take seeds and you genotype them, the mom is not the dad, they have some other dad. And like, I never saw a bee. So I'm absence of seeing pollinators doesn't mean they're not out there. So yeah, but so I think there is but they're pretty big bumblebees that can move far. Yeah. Go ahead.
Pretty quick. The pin 5 tandem duplicates
Yeah.
seems like an obvious kind of target for something wrong. Have you compared the sequences, the parallels to see?
Yeah, so everybody has the pair.
Oh, okay.
Yeah, sorry. Yeah, so no, it was like, because almost everything we study and else not in the system ends up being a copy number variant. So I'm very much like, oh yeah, you're gonna get down there and it's gonna be like, yeah, presence, absence, polymorphism versus, yeah. So yeah, but as far as, and I should say, and actually that pin 5, I'm starting to think it's only root expressed, does both of them as far as I can tell. So it doesn't mean it can't cause the of ground meristems to do different things, because certainly auxin signaling is complex, but it's not like as good a candidate as it was now that we have expression level. But it's, on the other hand, yeah, I hesitate to rule anything out. It could be all of them. Yeah, but yeah, almost everything else is a common variant, all the flowering type stuff. Yeah.
I was just curious, since both phenotypes are so controlled by their environment, how you think that future changes with climate change to that region?
Yeah. So I am not worried about Mimulus guttatus as a species or a species complex in the face of climate change, because between the Aleutians and Baja and Yellowstone and everywhere else, and even just our population in Iron Mountain is like more variation than there are between humans and chimpanzees. So there's plenty of variation for a variety of things. If there stops being snow, at which point things will be very bad, because there's like usually 5 meters of snow every winter in Yellowstone. If there's no snow or not enough snow for the plants to even, and they can always keep, I'm pretty, if there was no snow, they were toast. Yeah, but then at that point, Western North America is pretty well done without snow. I think you're absolutely right. It's a very dynamic situation in that even I think year to year, the snow levels vary. So we did 2 years of intensive field work in that one site, and the dry down was about a month different. So that's just normal. That was like 2010, 2011, where Margaret was out there like every couple of weeks, like snowshoeing to get through the winter, because we can't, even though the site's flowering, we have to get through the snow to get there. So I think there's a lot of variation, and I think actually that's kind of good news. This is also what we see up at Iron Mountain, where we study sort of just within population variation, is that having the history of temporal variation is useful when the bigger changes come, because at least if you're Minos Catatus, there's a lot of standing variation that is pre-adaptive for more extreme directional change. So it's not to say that, again, like with our site in Iron Mountain, it's also snowmelt fed. If there stops being enough snow, that population will go extinct, but all of its variation is out there on the landscape already. So I'm not too worried. With HQT, it's a little worrying that that's the one population that has been actually named as a different species, Aurothornthe thermalis. And so technically it's like an endangered species because it's one hillside, it's inside a national park. I'm not too worried about it, but that one, because it is strictly snowmelt driven, and it does have some variation that's kind of unique to it that I didn't really talk about today, that could wink out if there's not enough snow for it to actually get to reproduction. But I think there, some of those plants are making like 30 fruits. And so if the growing season was shorter, they might make 10 fruits, but it's a while before they make no fruits. Yeah. Okay, thanks.
Thank you. If anybody has questions, feel free to meet us.